Hello everyone, and welcome to today's presentation. I am excited to guide you through this session on Class 12 Biology, strictly based on NCERT. Today, we're focusing on a very important topic, reproduction in organisms. This is the first slide, so I just want to give you a quick overview. We'll cover the fundamental concepts of how different organisms reproduce, the mechanisms involved, and why reproduction is vital for life. So, let's get started and explore this fascinating subject together. Reproduction is a biological process by which an organism gives rise to young ones, or offspring. This is how living things create new members of their species. Offspring are usually similar to their parents, which helps ensure that the characteristics of the species are passed down from one generation to the next. This process is essential because it ensures the continuity of the species, allowing life to persist over time. You might also find this concept mentioned in NCERT Chapter 1, as it forms the foundation of understanding how living organisms propagate. So, in simple terms, reproduction is the key process that keeps life going and maintains the diversity we see in the natural world. Let's talk about lifespan, which is essentially the period from the birth of an organism all the way to its natural death. One interesting point to note is that there's no clear link between the size of an organism and how long it lives. For example, a butterfly might only live for about 1 to 2 weeks, while a crow can live up to around 15 years. And then, we have some remarkable cases like parrots, which can live up to 140 years. This diversity really highlights how lifespan isn't simply determined by size, but rather by a complex mix of biological and environmental factors. Understanding these differences helps us see the incredible variety of life on Earth and how evolution shapes these patterns. Now let's look at the two main types of reproduction as classified strictly according to NCERT. First, we have asexual reproduction. In this type, a single parent is involved, and the offspring are essentially identical to the parent. This process is usually quicker and requires less energy, which is why it's common in many simple organisms. Next, we have sexual reproduction, where two parents contribute genetic material to produce offspring. This leads to variation, which is important for the evolution and adaptation of species. These two types of reproduction form the foundation of how organisms propagate and ensure the survival of their species. Understanding these basic differences helps us see how diversity in the living world is maintained. So let's talk about single-parent involvement in asexual reproduction. In this process, only one parent is needed, which makes it quite different from sexual reproduction that requires two. Interestingly, there's no formation of gametes, meaning no egg or sperm is involved at all. Because of this, the offspring are genetically identical to the parent, essentially clones of the original organism. This allows for rapid multiplication, which can be really advantageous in stable environments where the parent's traits are well suited for survival. Overall, this method of reproduction highlights the efficiency and simplicity of asexual reproduction, and it plays a significant role in the broader context of how organisms can quickly establish new populations without the need for a partner. Let's explore the various modes of asexual reproduction, which are crucial for understanding how organisms can multiply quickly and efficiently. First, we have binary fission, a process where a single organism divides into two identical daughter cells, commonly seen in bacteria. It's a simple yet effective method of reproduction. Next is multiple fission, where the nucleus divides multiple times before the cell splits, producing several offspring at once, often seen in certain protists. Then there's budding, where a new organism develops as a small outgrowth or bud on the parent, which eventually separates. This is typical in yeasts and some invertebrates. Fragmentation involves an organism breaking into parts, each of which can grow into a new individual, a common method in some worms and algae. Vegetative propagation is especially interesting because it allows plants to reproduce asexually through parts like stems, roots, or leaves, leading to new plants that are genetically identical to the parent. Lastly, spore formation involves the production of spores, durable, resistant cells that can survive harsh conditions and grow into new organisms when conditions are favorable. All these modes highlight the diverse strategies organisms have evolved to reproduce without the need for sexual processes, which ties back to the overall theme of how life adapts for survival and proliferation. Common in amoeba and paramecium, binary fission is a fascinating process of asexual reproduction. The first step is the division of the nucleus, which is crucial because it ensures that each new cell will have a complete set of genetic material. Once the nucleus has divided, the process continues with cytokinesis, where the cell's cytoplasm splits, forming two separate and identical cells. This method is efficient and allows these single-celled organisms to reproduce quickly, maintaining their population. Understanding binary fission helps us see how simple organisms like amoeba and paramecium can rapidly multiply, which ties into the broader theme of how life can sustain itself even at the most basic levels. In this slide, we're looking at budding, a form of asexual reproduction seen in organisms like yeast and hydra. What happens here is that a small outgrowth, often called a bud, begins to form on the parent organism. This bud gradually develops and grows, almost like a tiny copy of the original. Once it's fully developed, it detaches from the parent and then grows into a completely independent individual. Budding is particularly interesting because it allows these organisms to reproduce quickly and efficiently without the need for mating. It's a great example of how different species have evolved various methods to ensure their survival and propagation. This process ties into our overall theme of asexual reproduction, highlighting how some organisms can produce offspring that are genetically identical to themselves, ensuring continuity and adaptability in their environment. Now, let's talk about how new plants can grow from vegetative parts. These are parts of the plant like rhizomes, tubers, and bulbs that naturally develop into new plants. Rhizomes are underground stems that spread out and produce new shoots, while tubers, like potatoes, store nutrients and can give rise to new plants. Bulbs, such as onions, are layered structures that can also develop into new plants when conditions are right. In addition to natural methods, humans have mastered artificial techniques to propagate plants. These include cutting, where a piece of the stem or root is planted to grow a new plant; grafting, which involves joining two plants so they grow as one, often to combine desirable traits; and layering, where a stem is encouraged to develop roots while still attached to the parent plant, then separated to become a new plant. All these methods fall under vegetative propagation, which is a form of asexual reproduction. It's especially useful because it allows us to produce new plants that are genetically identical to the parent, ensuring consistency. Understanding these natural and artificial methods is crucial, as they form the foundation for many agricultural and horticultural practices. They help us efficiently multiply plants, improve crop yields, and preserve desirable plant traits. Today, let's look at how sexual reproduction involves two parents working together. It begins with the fusion of male and female gametes, which are the reproductive cells. When these gametes combine, they form a zygote, which is the very first cell of a new organism. An important outcome of this process is genetic variation, meaning each new individual has a unique combination of genes. This variation is essential for the diversity and adaptability of species. Overall, sexual reproduction plays a vital role in maintaining healthy and diverse populations, connecting back to our broader discussion of how living organisms reproduce and pass on traits. Let's talk about gametogenesis, which is the process of forming gametes, or reproductive cells. This process is crucial because it ensures that each gamete is haploid, meaning it contains only one set of chromosomes. This is essential for maintaining the correct chromosome number across generations. Next, we have gamete transfer, which is about how these gametes move from one organism to another, typically during reproduction. This transfer is vital for fertilization to occur. Now, focusing on the formation of haploid gametes, this involves meiosis, a special type of cell division that reduces the chromosome number by half. It ensures that when two gametes fuse during fertilization, the resulting zygote has the normal diploid number. Before fertilization happens, there are several important events known as pre-fertilization events. These include gamete maturation, where the gametes develop fully and are capable of fertilization, and the mechanisms that bring sperm and egg together, like copulation in animals or pollination in plants. These pre-fertilization steps set the stage for successful fertilization, which is key to ensuring genetic diversity and the continuation of species. So, understanding these processes helps us appreciate how complex and finely tuned reproduction really is. Now, let's look at the process where the male and female gametes come together. This is known as fertilization. Essentially, it's the fusion of these two reproductive cells, which can happen either externally, like in aquatic animals where eggs and sperm meet outside the body, or internally, inside the female's body, as seen in mammals and birds. The key point here is that this fusion results in the formation of a diploid zygote. This zygote contains a full set of chromosomes, combining genetic information from both parents. This process is fundamental because it restores the diploid number of chromosomes and initiates the development of a new organism. So, understanding fertilization helps us grasp how genetic material is combined at the very beginning of life's cycle. Let's now look at what happens immediately after fertilization. The first key event is zygote formation, where the sperm and egg fuse to create a single, new cell. This marks the beginning of this remarkable journey. Following that, we have embryogenesis, which is the process where this single cell starts dividing and developing into an embryo. It's a highly coordinated series of events that sets the foundation for all future development. As the embryo grows, it develops into a new individual, with complex structures and functions forming over time. These post-fertilization events are crucial because they transition us from a single fertilized cell to a fully formed organism. Understanding these steps helps us appreciate the incredible complexity and precision involved in human development. Let's take a closer look at the differences between asexual and sexual reproduction. In asexual reproduction, there's only one parent involved, which means the offspring are essentially clones of the parent, leading to no variation. This process is usually rapid, allowing populations to grow quickly when conditions are favorable. On the other hand, sexual reproduction involves two parents, combining genetic material from both. This results in offspring that have variation, which is important for adaptation and survival. However, sexual reproduction tends to be slower because it requires the involvement of two individuals and more complex processes. Understanding these differences helps us see how organisms reproduce in ways that suit their environments and survival strategies. Let's now look at the concept of clones. Clones are individuals that are morphologically and genetically similar to each other, essentially identical in their genetic makeup. This is often seen in organisms that reproduce asexually, resulting in offspring that are exact genetic copies of the parent. Moving on to the different life stages, organisms typically go through a juvenile phase where they grow and develop. Then enter the reproductive phase, during which they can produce offspring. And finally, they reach the senescent phase, where their biological functions decline. It's also important to understand the difference between seasonal breeders and continuous breeders. Seasonal breeders reproduce during specific times of the year, often synchronized with environmental conditions that favor offspring survival. Continuous breeders, on the other hand, can reproduce throughout the year without strict seasonal restrictions. And finally, from the NCERT perspective, understanding these points is crucial because they highlight how different species have adapted their reproductive strategies to their environments. Recognizing these patterns helps us appreciate the diversity of life forms and their survival mechanisms.